Good day, good and welcome back to another lesson. My name is Mpukui and I will be facilitating this lesson. And essentially, what we'll be doing in this last segment of the DNA Code of Life section, we'll be looking at protein synthesis. Now, boys and girls, what is to synthesize something? When we synthesize something, we make something. So we essentially are in a process whereby we are creating something. Now, protein synthesis, boys and girls, is a process in which proteins are manufactured or made in living cells. Now, this sophisticated process, boys and girls, occurs in what we call two stages. Now, the first stage of protein synthesis will take place inside the nucleus, and that stage is transcription of DNA. We are transcribing DNA. And the second stage is translation of RNA to the protein. So that will take place outside the nucleus and at the ribosome. So translation occurs at the ribosome, transcription occurs inside the nucleus. Two main stages, transcription and translation. You'd see that I underlined transcription and translation in red because those are the key words that are integral when asked in a question. And we would ask you at times, we'd say, boys and girls, now what is the significance? Why are we replicating? What is the process of, what is the significance or the function of RNA? RNA makes or aids or plays a role in protein synthesis. Now, why do we need protein synthesis? Why are proteins such an integral part? We will get to that point. I want you to think about this as we move along. Why are we making proteins? Why are we making proteins? Think about it. Okay. Now, that being said, I want us to then look at the process of transcription. We said when making proteins, protein synthesis occurs in translation and transcription. But what comes first? Transcription. We are transcribing. Okay. And I said it's very important for you to know your DNA replication process. Because when you know that, you'll be able to apply it here. And in the previous video, I also gave you guys I introduced you guys into the process of transcription and translation, and you'll see how easy it is for you to know transcription and translation, provided that you listened to the previous video. If you did not and you don't remember those processes, go back, because you're doing exactly what I said you should not do. You can't move on to the next video if you don't understand what's going on in the first video, and if you don't know if the questions posed in the notes were to be asked to you and you don't know how to answer, you can't move on. Because these are the foundations, these are the building blocks, these are what we'd call the monomers of this whole section. That being said, how do we transcribe proteins? Transcription occurs in the following way. DNA double helix unwinds, the double-stranded DNA unzips, right? And one strand, I want us to remember this, one strand is used as a template to form mRNA from free-floating RNA nucleotides in the nucleoplasm. So one strand is used in this case. mRNA is then complementary to DNA and has the coded information for protein synthesis, or has the coded message for protein synthesis. So how does transcription occur? Transcription, the DNA double helix unwinds. Remember, in proteins, in a process whereby we're making DNA, the DNA replication, the DNA double helix unwinds. The two strands then unzip. In this case, one strand is used as a template to form mRNA from the free-floating RNA nucleotides in the nucleoplasm. And then from there on, mRNA is complementary to DNA. It's complementary to DNA, how? I'll show you now, but we'll do an activity on that. It's complementary to DNA, and mRNA has the coded message for protein synthesis. Now, the process of translation. Translation, we are now at the ribosome. So what happens is tRNA carries specific amino acids, provided that an anticodon on tRNA matches the codon on mRNA, and it carries it and brings the amino acids to the ribosome. And now what happens? This is an easier part. Step 3. The amino acids are joined to each other by peptide bonds, right? And then when 50 or more amino acids are joined together, the required protein is manufactured or formed. I want us to quickly do this quick activity here. So we'll have first DNA, then mRNA, then tRNA. This is what usually happens in protein synthesis. For example, let's say we have adenine, we have guanine, we have cytosine, we've got thymine. In order for us to have the coded information for protein synthesis, mRNA, as said here, must be complementary to DNA. And how do you complement something? By giving it what we want. We need to lock hands. The lock and key method, we need to lock hands. So, okay, we said that RNA, we'll see we're dealing with mRNA. We said that RNA has multiple, multiple nitrogenous bases. We have guanine, we have cytosine, we have adenine, and we have uracil. We don't have what? We don't have thymine in RNA. So what would we have here? The adenine would bond to uracil. The guanine, cytosine. The cytosine, guanine. The thymine, adenine. Now, this is a stage whereby we're moving on. So the DNA double helix unwinded in transcription. The 2 strands unzipped. One strand was used as a template to form mRNA from free-floating nucleotides in the nucleoplasm. Then mRNA is then complementary to DNA. We can see how is it complementary. Adenine bonds to uracil. Guanine bonds to cytosine. Cytosine bonds to guanine. Thymine bonds to adenine. Now we move on. When this is done, mRNA will have the coded message for protein synthesis. Perfect. Now we're going to move on to tRNA. What happens to tRNA? Okay, we say that tRNA picks up specific amino acids in the nucleoplasm, provided that the anticodon on tRNA matches the codon on mRNA. So now how would we match? We would match by